Hello, my name is John Peters, and I am the product manager for elemental spectroscopy instrumentation at Shimadzu Scientific Instruments. In this webinar, I will introduce some tips and method techniques for sample preparation for elemental impurity analysis in pharmaceuticals. Top goals are to reduce contamination from the environmental and equipment sources, and to measure and validate analytical data for optimum accuracy and precision. So let's begin. Routine elemental impurities analysis of pharmaceuticals involves three steps: sample preparation, standard solutions preparation, and measurement via ICP-MS, inductively coupled plasma mass spectrometry. It is critical to ensure that these steps are conducted in a way that does not cause loss of elements in the sample due to volatilization or precipitation, etc., that there are no contamination from reagents or tools, and that the sample is stable enough through the time of measurement. Also critical are maintaining the sample introduction hardware and ICP-MS instrument, checking for signal drift, and ensuring proper performance of the instrument. First, I will go into some detail about sample preparation and preparation of calibration standards. Most pharmaceutical samples do not dissolve directly, even in strong acid solutions. Samples must be decomposed in pressurized microwave digestion vessels, usually made of Teflon. These vessels are reusable and hold the sample, acid, and other reagents for digestion. They are sealed and subjected to microwave radiation to produce heat and pressure, which renders the contents soluble in water while retaining the elemental compounds. Samples must be weighed accurately so that the end sample dilution is of known sample weight and in a known volume. This allows comparison between samples and standards, which are also prepared as weight to volume. Weighing samples into Teflon vessels can be tricky. Static electricity causes powders to disperse. Transferring weighted samples to the bottom of the digestion vessel is critical for accurate analytical results. Powder that adheres to the top of the vessels may not digest in acid or end up in the sample dilution, even though they contribute to the weight of the sample. Spatulas made of resin are recommended for this, along with an ionizer that helps reduce static electricity. Spatulas and the vessels should be rinsed with acid ahead of sample preparation to remove contamination. The quantity of sample depends on digestion recipe, but is often about half a gram of material. After sample is accurately weighed into the digestion vessel, acids and other reagents required by the digestion recipe are also added. Water should be trace elemental grade, greater than 18 megalom. Acids such as nitric, hydrochloric, hydrofluoric, or hydrogen peroxide should all be trace metals grade, not ACS grade, and none that are packaged in glass. Ultra-pure trace metal grade for electronics and semiconductor industries offers the lowest level of contamination, PPT levels of impurities, and are recommended for the best possible detection limits in your method. Any background will increase the signal-to-noise ratio in your blanks and affect the method detection limit calculations during method validation, determined by measuring multiple method blanks and calculating the standard deviation. Method blanks are prepared by adding the same quantity of reagents minus any sample into their own digestion vessels. This allows for calculation of how much elemental impurities are contributed to instrument signal and analytical results from the sample preparation process and the instrument. To further reduce contamination, it is recommended to rinse pipette tips with clean acid before adding reagents to the digestion vessels, and to use clean digestion vessels. The quantity of reagents is dictated by the digestion protocol, which is specific to each sample matrix and is available from the instrument manufacturer. Generally, it is recommended that a small amount of water is added first, and then the acid, then venting the solution in a fume hood until it stops reacting with the acid. After this, the remaining water is added, the vessel is sealed, and the digestion protocol is initiated. Next, some comments about the digestion vessels themselves. Contamination and carryover from digestion vessels is the most common source of inaccurate and false positive results in pharmaceutical trace elemental analysis. Cleaning the vessels thoroughly is a critical part of a validated process. Vessels are cleaned regularly with strong acids. However, they should also have regular cleaning cycles on the digester. This would involve adding acids to the vessel and running it through a digestion protocol recommended by the manufacturer. Solutions from this process can be tested via ICP-MS to check if the vessels are clean. If the method blanks are unusually high, this is a good first step in eliminating environmental contamination. Disposable vessels can also be used with some digesters to eliminate this source of carryover contamination. However, if reusable Teflon vessels are used, it is recommended to clean them regularly and to measure high concentration samples in a separate designated vessel from low concentration samples. After digestion, vessels are removed from the digester and allowed to cool. Opening the tops of the vessels slowly allows venting of the gases without losing sample. Transferring the liquid remaining in the digestion vessel to a volumetric flask and rinsing multiple times with ultra-pure water, then bring to volume with water. This ensures all the elemental material from the weighed sample is transferred to the known volume of the dilution flask. Avoid glass materials at all costs. Glass both absorbs elements of interest like arsenic and mercury, but also contributes to contamination of multiple elements of interest. Cleaning reusable volumetric flasks is another critical component of a validation process. New containers or disposable volumetric flasks should also be cleaned prior to use. Examining the digestion is a critical step. The material that is produced by the digestion process should be clear and colorless, but may be light yellow color. Dark yellow or solid material or any turbidity in the cloudiness of the solution indicate incomplete digestion. Revisiting the digestion protocol is recommended, and adjusting the acid and reagent composition, the sample weight, and/or the temperature ramp. It may require higher temperature or longer hold times or more and different acids. Some samples, particularly plant material, will have silicate particles floating in an otherwise clear and colorless solution. These can be removed by filtering through a 0.45 micron syringe filter, as the particles will clog the nebulizer and cause signal drift. Hydrofluoric acid can also be used to dilute and digest these samples, but filtration offers many advantages, not the least of which is safety, particularly if silicone is not an element of interest in the method. Next, I will discuss preparation of calibration standard solutions. Single-element or multi-element certified calibration standard stock solutions are available from many reputable commercial sources, and using these saves a great deal of time preparing standards from raw materials. Dilution of these stock solutions is relatively straightforward. A point of consideration is matrix matching, ensuring that the sample dilution and the standard calibration solutions have the same acid matrix. So, for example, a typical digestion with 4.5 milliliters of nitric acid and half a milliliter of hydrochloric acid diluted to 100 mils, or 50 mils, and then a 1-to-1 final dilution, would have a 4.5% nitric acid and 0.5% hydrochloric acid matrix. The standard solution should match this. To prepare standard dilutions, stock solution is added by a pipetter in different volumes and diluted into volumetric flasks. Dilutions should be prepared regularly. and the stability time of standards should be validated by measuring single stability over time. Different elements are stable in different acids, but generally for pharmaceutical analysis, nitric acid with 0.5% hydrochloric acid to stabilize mercury works for a wide range of elements and sample types. Units of the standard dilutions are in weight per volume, the same as for sample preparation. Multi-element standard stock solutions are available commercially from a variety of sources and can save a lot of time preparing calibration standards. Many common ones related to USP 232 are pictured here. For additional details, see the website of each reagent manufacturer. In addition, there are mixed standard solutions specific to unique oral formulation requirements and other USP requirements, each sold by Millipore Sigma, Spec, Certiprep, Inorganic Ventures, High Purity Standards, and other manufacturers. So, having discussed sample preparation, digestion, the standard prep, now I'd like to go into some detail about measurement operations on the ICP-MS. First, some overview about ICP-MS operation. First, the plasma is lit and the instrument allowed to warm up. Then, sample conditions can be evaluated. Measurement conditions can be optimized through instrument tuning, and calibration curves can be run. The calibration curve linearity, sensitivity, and precision are verified. Accuracy is verified by running confirmation tests. Continuing calibration verification samples, which are standard solution dilutions, run as unknowns. Blank samples are also run to determine limits of detection, signal drift, carryover, contamination, and overall performance of the instrument. A bit more detail about instrument and environmental contamination considerations. Contamination of samples during preparation is one source of false positives in trace elemental analysis. However, environmental contamination is also possible. We recommend taking measures to ensure cleanliness of the samples after they are placed on the autosampler for analysis. Dust from the atmosphere can be prevented by installing an autosampler enclosure, which uses a fan or vent to move air through a HEPA filter into the sample chamber. This removes acid fumes from the work environment, but also prevents environmental dust from contaminating samples. Different autosampler designs are also capable of controlling movement functions such that the probe never travels over samples that have not been analyzed already. In this way, any droplets that fall off the sample probe during movement fall into a tray and not into samples. This is particularly important with CCV samples, where the probe goes into the same vial multiple times and travels past other standard vials on its way to the rinse station. Programming in XYZ design autosamplers, such as a CTAC ASX280 or ASX560, allows the probe to travel over no samples that have not previously been run, and reduces potential contamination. Generally speaking, the cleaner the environment, the less possible contamination will be likely in your analysis of pharmaceutical samples. Before the measurement is carried out, the sensitivity of the instrument should be checked along with signal stability. After the sample and internal standard solutions have come to room temperature, nebulize each and check the movement of bubbles through the transfer tubing. It should be consistent and smooth. Otherwise, the peristaltic pump tubing should be replaced or the pump tension arm adjusted. The time it takes to get sample from the tip of the autosampler probe to the plasma should be verified, as this information is necessary to program an effective uptake time and rinse program into the method. For instrument verification, run an instrument validation and instrument tune. If this adjusts various instrument parameters such as lens voltages and torch position for optimum performance. Regular periodic inspection and cleaning of consumables is also necessary for optimum performance: torch, cones, nebulizer, and pump tubing. If signal is low, consult the help guides with your instrument, but common issues include clogging of the nebulizer. Aerosol performance can be observed by nebulizing water into air and observing the spray, which should be an even fine mist. Uptake rate can also be measured via displacement using a stopwatch and a graduated cylinder. The recommended uptake rate for most nebulizers can be found in the specifications sheet and can be altered by changing the pump speed or the nebulizer gas flow. Cleaning and backflushing the nebulizer is recommended regularly and to fix uptake problems, and can be accomplished easily using a special tool. All glass parts can be cleaned with strong acid and soaked for extended periods, but do not sonicate glass. Cones should also be inspected. Any deterioration of the material surrounding the orifice or blockage from deposited solids should result in cleaning and/or replacement of the cones. Cones should be soaked for a short period of time in dilute acid, but can be sonicated. Condition them by nebulizing pure water or 5% nitric acid after cleaning and drying. Cones can also be conditioned with 30 minutes nebulizing acid solution with 100 ppm sodium or calcium, whichever analyte is not in the method. Pump tubing can become deformed, particularly if cavitation happens after the instrument is shut down for the day and the tension of the pump arm is not released. This is addressed on the Shimadzu ICP-MS 2030 by continuing to rotate the pump after the sequence is complete and automatic shutdown of the plasma occurs. The pump now turns backwards so as not to draw up internal standard solution overnight, saving solution and the pump tubing. The torch should be inspected for vitrification, cracks, and salt deposits. Regular cleaning should be employed, depending on the acid matrix and salt content of the samples. Soaking in nitric and hydrochloric acid is preferred, but again, do not sonicate glass. Regarding the calibration curve, after programming the calibration samples into Lab Solutions ICP-MS software and measuring the standard solutions, check the points. If contamination is present, blank sample intensities will show signal. If there are problems with the sample introduction system, RSDs of each sample reading will be high. The correlation coefficient for the calibration curve, the R value, should be greater than 3 nines. If it is not, preparation of the standards may be required again. You can look at multiple elements in a calibration curves and see whether or not they follow the same pattern for additional information about what the source of the problem could be. Check also that the lowest point on the calibration is discernible above the background blank signal intensity. Lab Solutions ICP-MS software calculates the 3 sigma limit of detection and also the 10 signal, the limit of quantitation, from the calibration curve to approximate LOD and LOQ for the method. Validation, however, should consist of running calibration curve and then more than 6, up to 20 is recommended sometimes, method blanks, and then calculating the standard deviation of signal intensities from these. The error or standard deviation of the blank sample should be less than 1/10 of the target concentration, or J, which is ideally 1/10 the lowest permissible concentration in the samples from the permissible daily exposure levels specified in the regulations, either USP or another. More on this later. Here are some notes on different signal drift situations and their possible causes. Figure 1 shows internal standard signal that is stable between samples. This is ideal and indicates no problem with the sample uptake or instrument performance. In Figure 2, we see gradual and steady decline in signal intensity. Multiple causes of this could be occurring, including memory effect from the previous sample or progressive clogging of the nebulizer by particulates. Memory effects can be addressed by changing the rinse solution on the autosampler, increasing the amount of time the system is rinsed between samples, or the rate of sample rinse between samples. Clogged nebulizers can be remedied by backflushing and cleaning, then by filtering out particulates from the sample matrix. In Figure 3, we see gradually increasing signal intensity. In this case, it may be that not enough time is programmed into the method for sample uptake. You will see a dramatic increase in signal intensity for analysis if this is the case and the instrument is collecting signal data before sample reaches the detector. If this is not the problem, examine the peristaltic pump tubing and mixing tee to make sure internal standard is present and the uptake from both probes is consistent. The probe tubing may need to be replaced, or the mixing tee cleaned, or the connections secured for leaks. Note that internal standard fluctuations and sample signal fluctuations can mean different things, and so both should be examined. Number 4 shows random distribution of unstable signal. Verify that there is instrument sensitivity by checking a different sample with known concentrations, like a standard. Sporadic signal intensity is expected when concentrations are very low, but if there is analyte in the sample and sporadic sample intensity is still observed, in this case the limit of detection is likely poor, the instrument sensitivity is likely low, and it is recommended to tune the system to manufacturer's protocols and check uptake hardware. Signal intensity is one of the parameters checked in instrument validation and daily performance checks, and various remedial actions are detailed in the help sections of the instrument operating guide. So, how sensitive does the method need to be? Permissible daily exposure levels are detailed in USP and other guidelines and are generally stated in units of contaminant weight per day. Assuming a daily dose of 10 grams per day and applying the rule that we need to quantify to 10% of the lowest permissible level, we see the units change to micrograms per liter, weight over volume, and various concentrations below and above the J value calculated. The carryover signal and method blanks must be lower than 0.5 J. In this case, we see that nickel has a higher PDE value than cadmium, so the calibration curve concentrations and required sensitivity for each is different. That said, while the LOQ may be higher for elements like nickel, removing contamination may be more difficult with abundant elements than ones that are uncommon in nature. The chart here shows levels of hundreds of ppb range, which is well within the sensitivity of ICP-MS when performing correctly. Other technologies for elemental analysis do not have this level of sensitivity, so ICP-MS remains the standard for pharmaceutical samples analysis in trace metal contamination. We mentioned that the memory must be lower than 0.5 J value calculated in the previous slide. The way to check this is to run the highest calibration point and then a blank sample afterward. The signal ratio after the high concentration sample that carries over into the subsequent blank is the ratio of signal intensities. Mercury is a common difficulty for carryover. Stabilization of mercury with 0.5% hydrochloric acid is strongly recommended in the samples, calibration standards, and rinse solution. 1 ppm gold can also be added to good effect, but if full mass scan is turned on for the detector, it will shorten the life of the ICP-MS detector by constantly measuring high gold intensity. Memory effects can also be reduced by having multiple rinse solutions available in some autosamplers and having different solutions in each, dissolving organic and difficult inorganic ingredients separately. We can also lengthen the rinse time in the method and speed up the rinse solution uptake. Ideally, the autosampler should efficiently rinse both the outside of the probe and the internal tubing all the way to the nebulizer. A good rule of thumb is that 1/10 or less carryover from the highest calibration standard should be expected. Checking the blank samples is also required to ensure that signal is lower than the 0.5 J value. If the method blank is high, it means that the conditions are not producing sufficient sensitivity for acceptable analytical results. Method blank contamination techniques previously discussed should be employed to clean the vessels and sample introduction equipment before trying again. After confirming contamination is low, sensitivity is high, and carryover is acceptable, accuracy of the analytical results are also examined. Some points to consider here as well. Variation in the sample should be low. 1% RSD is ideal and possible with higher concentration samples. Less than 3% is good for most sample matrices, and most SOPs have an acceptable RSD limit of 5%, especially at low concentrations. This is with multiple scans of a sample nebulizing continuously. The RSDs can be changed by changing the integration time or the number of scans, while this does affect the analysis time. Drift should also be checked, as it was in previous slides. Accuracy should be checked by measuring samples of known concentration against the calibration curve. A standard dilution run as an unknown will produce a number ideally within 20% of the expected value. A summary of analysis results can be viewed here. We checked the calibration curve calculated at concentrations around the J value from USP PDES. We checked memory effects in our method and method blanks to ensure sensitivity and absence of contamination. Calibration check standards are also added periodically during analysis of unknown samples so that the accuracy of the system is monitored continuously. Not pictured are QA/QC functions where internal standard signal drift is monitored, and any drift here or in the CCV can trigger a recalibration or stoppage of analysis for troubleshooting. So, to summarize, we recommend sample preparation, digestion, environmental, and sample introduction hardware systems to be optimized for low contamination potential. Contamination, drift, and variability can be quantified easily during analysis. Calculating the target concentrations can be done easily using the relevant regulation, USP or otherwise. The monitoring levels 10 times lower than this after converting to concentration units with a known daily consumption rate. In this manner, an analytical SOP can be validated, data defended effectively during any regulatory agency inspection of your laboratory. We look forward to assisting you with any implementation of these techniques and answering any questions. We appreciate your time today and welcome comments and questions. I will now turn the meeting over to our moderator. Thank you again.
Hello, everyone. Thank you for joining the Shimazu webinar for ICP-MS analysis for elemental impurities in pharmaceutical products. My name is Haihan Chen. I'm a product coordinator in elemental spectroscopy group at Shimazu Scientific Instrument. I will moderate the Q&A session today. So if you have any questions, please type your questions in the Q&A window on the right of your screen. We will try to cover as many questions as we can, but if we are not able to cover your questions live, we will send the response through email. So please feel free to type in any questions you have. So John, I have one question from a customer. Can other pharmaceutical metrics like tablets, gels, and liquids be measured by ICP-MS?
Yeah, thanks, Haihan. The answer to that is yes, assuming that it's possible to digest that matrix. Most things like tablets and gel caps and powders and things like this digest pretty well through standard protocols on a entry-level digestion system like the Mars 6 or the Multiwave Go. Some that may have more silicates or are more difficult to digest may require a higher-end Multiwave 7000 or higher temperature digester to get it into solution. But the limitation is more about getting the material dissolved and digested rather than the ICP-MS. Once it's in solution, the ICP-MS can handle anything. But great question.
Okay. So the next question we have is, taking into account the complexity of some metrics in different drugs, which internal standards do you recommend? Like the customer has issues with scanning as an internal standard.
Yeah, that's a good question. So we published some guidelines for pharmaceutical analysis, and we do sometimes see if the matrix has a great deal of calcium in it, that that there's some transfer from the element to the side of that scandium, and so some carryover that can affect the internal standard if there's a high amount of calcium in the matrix. So in those cases, we recommend germanium, but we have other options as well, depending on what the matrix is and what types of contaminations are likely to be present in that matrix. But we can certainly provide the recommended guidelines that we published, and can answer a lot of questions in more detail than I have time here. Yeah, good question.
Good. So the next question is, if there's a repeatability problem, what could be the reason for this? Like arsenic, mercury, selenium, what would be your suggestions to resolve the repeatability problem?
Sure, that's a great question. The first thing I would look at is whether or not the internal standard signal is stable or not. If that's jumping around, then it's a sample introduction issue, something with the peristaltic pump or the nebulizer, etc. If the internal standard is stable and it's the signal that's jumping around, that tells you some different information. And we did go into some detail about this in looking at the slides here. Slide number 17. It depends also on what type of variability you're seeing. If you're seeing a gradual decrease in the signal and that's causing RSDs to be high, or if it's rising versus if it's completely scattered, tells you different information, and dictates the approach to fixing that problem depending on what you're seeing. So for example, if it's decreasing gradually, that's often a clogged nebulizer that's restricting the uptake of sample, which can be fixed by backflushing the nebulizer, changing the aerosol pattern by making sure that everything is clean and that the uptake is correct, etc. So different techniques depending on which problem you're seeing. But the first thing is to look at whether it's the internal standard that's also fluctuating or if it's only the sample itself.
Okay, good. So the next question from the customer is, according to your experience, how fast USP is adopting the use of ICP-MS against AA?
I think USP is considering ICP-MS to be the standard, the gold standard. AA is fine, but the number of analytes that you can test semi-simultaneously on the ICP-MS makes it superior for a lot of reasons, throughput being the main one, and cost of analysis, but also the sensitivity. Even with graphite furnace, the sensitivity of the ICP-MS is much higher. So USP 232, 233 is the gold standard for pharmaceutical measurement.
Okay. So the next question from customer is, how does ICP-MS compare with other techniques like XRF for pharmaceuticals? How about ICP-E?
Oh, great question. So somebody just asked about the AA. Good sensitivity with the graphite furnace, but you can only measure a certain number of elements at a time. So it takes a long time to do a single sample with all of the elements required by the USP guideline. But something like XRF and Shimadzu makes a really good benchtop X-ray fluorescence instrument that can also measure all of these elements. The limitation there is detection limited. The sensitivity isn't quite as good as ICP-MS. So you can use it as a screening tool, and in most cases to test oral impurities, where the guidelines are much higher concentrations than inhalation, for example, or as a screening tool, or you send it out to a third-party lab. And because XRF is much faster and there's no waste, no sample prep, et cetera, you can get pretty good results very quickly. But the gold standard remains ICP-MS just because of the sensitivity.
Good. So the next question we have is the reaction cell required for elemental analysis for pharmaceuticals?
Yeah, that's a great question. We get this a lot. This triple quad and reaction cell are two features that help reduce isobaric interferences. That's where two different things have the same mass to charge ratio, either different isotopes or polyatomics, etc. So you can get more sensitivity for difficult elements of analysis using one or both of those techniques. The USP 232-233 guidelines don't have any of the elements that are difficult to analyze and that require a reaction cell or a triple quad. So a single quad ICP-MS with collision cell to remove polyatomic interferences is sufficient for pharmaceutical applications. Using a reaction cell is very handy, in particular with elements like phosphorus or sulfur. You can react the sulfur with hydrogen, for example, and change mass to charge ratio from 31 to 35, or you can change it into PO plus, so mass charge ratio 47, and measure that. And the reason for that is because phosphorus has a very high ionization energy, and so not much of it is converted into an ion. If you do change the plasma conditions to where you're ionizing phosphorus, you're also creating a lot of double charged interferences and high oxides, all kinds of problems for your method. So the standard conditions on an ICP-MS mean that you're not going to see very much phosphorus or sulfur, and the detection limits are poor. But if you have a reaction cell and you can turn it into some combination with hydrogen or oxygen or ammonia, then you can see it much easier and get much better detection limits. That said, phosphorus and sulfur are not elements that are required to report for US. It's good for some applications, but not required.
Okay, good. So the next questions we have is to analyze nutrients in plant. Is digestion required? No, digestion only for heavy metal analysis?
A short answer is yes, and the reason is that ICP-MS works by nebulizing a liquid sample into a glass chamber, and that chamber creates there's a fine mist that's pushed into a plasma. So the sample itself has to be in liquid form. That's the digestion for. That said, there are some interesting studies, again, using the XRF, the benchtop XRF, that don't require digestion. You can directly analyze a sample without doing any kind of processing. And that way you can look at different parts of the plant and see where the nutrients are being metabolized on that piece of material, whatever it is. So yeah, if it's plant material and you're looking at uptake of molybdenum from the soil or something, you can see where it's being deposited on the leaf or the stalk of the plant without digesting it. That said, again, the limitation is ppm level detection limits are common on the XRF, whereas you can get parts per trillion ICP-MS.
Good. So the next question is about cones. So what kind of cones, what kind of sampling cones and schema cones does Shimadzu provide what kind of cones would you recommend for pharmaceutical?
That's a great question. So we have copper cones, we have nickel cones. I prefer the nickel cones because they last a little bit longer and they don't contribute a lot of background to nickel sensitivity in the analysis, so it's fine even for PPT level nickel analysis. But if you're really trying to get very, very low levels of detection limit for nickel, the copper can be fine. They just have to be replaced a little bit more often. They're more sensitive to acids, and so the orifice dissolves a little bit more than nickel. But the best option is platinum cones, and my opinion is that over the course of the lifetime of an ICP-MS, after you replace a bunch of cones on a regular basis, the cost of that really adds up. Whereas our vendor for platinum cones currently will refurbish them for free. So you buy 2 sets of platinum cones, and that's thousands of dollars, but then you never have to buy cones again. They just continuously refurbish ones while you're using the second set. So much more cost effective in the long run, and you're going to not have any interferences in background from copper or nickel.
And the next question is about contamination. So how to avoid the contamination to arsenic, cadmium, lead, and mercury?
Yeah, great question. We went into some detail about how to avoid contamination in general in the webinar, and finding where the contamination is coming from by doing method blanks. But the best way to keep that from happening is to not use any glass. Use clean reagents, clean water, clean tools, and then to check your method blanks on a regular basis. The other thing that's important is to stabilize mercury and arsenic. They tend to be volatile and also very sticky, so you'll get carryover, and then you'll lose some of the analyte that's in your sample, just sticking to the sides of the vessel if it's not stabilized. And so because of that, we recommend 0.5% of hydrochloric acid to stabilize both of those. Gold can also be used. People use 1 ppm gold in their matrices, but that can be used in combination with 0.5% hydrochloric acid as well.
Good. I think that's all the questions in the Q&A windows. Unless there's no more, if there's no more questions coming in, we are going to end this section. Again, thank you for joining the webinars. And if you have any questions or anything we can do to support your running with your ICP-MS, please let us know. Thank you.
Yeah, thanks everyone.